In recent years, you have probably heard of the concept of gene editing. But do you know what it is? Gene editing is the use of biotechnological techniques to make changes to specific DNA sequences in the genomes of living organisms. This process is what allows material to be added, removed, or altered at particular locations in the genome, also called loci. In order to have a full understanding of gene editing, we must first start with the basic structure of DNA. DNA is a long molecule that contains our genetic information. It is composed of 4 nucleotide bases: adenine, cytosine, thymine, and guanine, which are essentially the genetic alphabet. These bases function in pairs, called base pairs, where adenine and thymine pair up, and cytosine and guanine pair up on separate strands to initiate the processes that give instructions to the cell. There are approximately 3 billion base pairs in the human genome. A genome is an organism's complete set of DNA. Sometimes there is a variation or change in the proper order of base pairs, called a mutation, which can often result in undesired phenotypes. Contrary to the everyday use of the word mutation, it is not always a bad thing. It is simply a change in the order of the genetic alphabet. Gene editing allows scientists to reverse or alter mutations in a way that instructs the resulting gene to code for the desired function, which could entail turning off the gene altogether. This results in a change of phenotype, which can mean a change in appearance, behavior, or any other observable physical and biochemical attributes. So, how exactly can genes be edited?
Engineered nucleases, which can also be thought of as molecular scissors, are enzymes that move down DNA strands and make specific double-strand breaks at desired places on the genome. They are made up of 2 parts, one to guide the nuclease down the DNA sequence, and one to cut the DNA. CRISPR, which we will discuss in a few moments, is a type of engineered nuclease. Once the desired section is cleaved, the DNA will naturally repair the cut by filling in the space once the uncleaved strand finds a nearby homolog as a reference for what should be there. However, the desired sequence will be introduced into this space instead of the previous sequence with the undesired mutation. Once repaired, the cell then continues to function but with new instructions. As a result, changes such as gene knockouts can be made, which help validate antibodies for receptor-specific binding. But this process can have flaws, such as mistargeting of the site to be cut, which can manifest by impairing the gene's ability to function properly or at all. An especially prevalent way of gene editing is with CRISPR.
CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. It is actually found in nature as segments created when bacteria capture snippets of DNA from microbial invaders and viruses. Its mechanism of action is through nucleic acid repair and metabolism. So, what does this look like in prokaryotes?
When bacteria detect the virus, 2 strands of RNA are created, one which matches the sequence of the viral DNA. The 2 strands form a complex with a nuclease called Cas9, which can cut DNA. When the matching sequence finds its target on the virus's genetic code, Cas9 cuts the target and disables the virus. The complex will lock onto the sequence, unzip the DNA, and match it to the target RNA. When complete, Cas9 will use 2 molecular scissors to cut the DNA, which will try to naturally repair itself. This repair process can be used to introduce the desired sequence into the cleaved space where the mutated sequence was previously. Eventually, scientists realized that Cas9 can be used to not only cut viral genes, but to cut any DNA sequence at a location of their choice by changing the guide RNA to match the genome of the target. There are other subtypes of CRISPR, like Cas3, which is helping to find ways to combat and manage antibacterial resistance. Cas13a is helping in the development of Sherlock, which stands for Specific High Sensitivity Enzymatic Reporter Unlocking, a monitoring and diagnostic system that could help with bacterial and viral outbreaks and potentially even cancers. The ability to edit the genome of these bacteria bears significant implications across many fields and topics. Genome editing can be used for anything from disease treatment to drug development to even crop science and agriculture.